Hello, everyone. My name is Zhang. I'm the Deputy Manager at LMB Flow Cytometry Facilities. Today, I'm going to give you an introduction to flow cytometry. So I'm going to talk to you about who we are and what we do, and what is flow cytometry, and how does the flow cytometry work. And I'll go on to some sample preparation and most importantly, the flow cytometry applications in biology. So this is our team, and we are located in third floor, 3N185 and 3N180. So there are 3 people in the team at the moment, and Piandri Patila is the manager, and I'm the deputy, and we have Chen joined us last year. We are currently recruiting the 4th person. And what we have in the lab, so there are a range of the flow cytometry instruments, mainly 2 categories. One category is the cell analyzers. The other category is cell sorter. So we train users on the cell analyzer. You use the analyzer yourself, and you book our time for the cell sorting. So there are about 6 cell analyzers in our lab. So if you go to our internal website and under flow cytometry, fluorochromes, and you can find the specification for each instrument we have here. On the table, we'll let you know the what lasers on that instrument, also how many channels we have in each instrument, and what kind of a color you can detect in that particular instrument. So we have 2 BD LSR for TESA, and one of it is equipped with 4 lasers, can look up to 16 color. The other one is 5 lasers, including a UV laser, can look up to 18 color. So we have BD LSR2, and this is a 4 laser instrument with 14 color detection. Again, you can see on the table, or we can what kind of color you can be detected on this particular instrument. So we have Beckham-Coulter Cytoflex XL. This instrument is equipped with 6 lasers, can look up to 21 colors. Also, this instrument is particularly good to look at the small particles. So we have a 1st generation of Sony spectral analyzers. This is a 3 laser 34 PMT detection system. And I will talk about a little bit more about spectral analyzer later in this talk. What is the difference to the conventional flow cytometers? So we have acquired Sony ID 7000 spectral analyzer, which will be installed on this Friday. So this instrument will give us 6 lasers. And this is up to 184 PMT detection. This really increased the capability of larger panel especially for the immunophenotypings. So we have 5 cell sorters in the lab, 3 Synergy high-speed sorter, and 2 of them are equipped with 5 lasers, can look up to 18 colors. And one of the Synergies has 3 lasers, look up to 10 colors. Again, please contact us for cell sorting. We can assign you to a particular instrument that you need for your sample. So we also have a BD Fax Aerial Fusion. This is a 6 laser system, look up to 18 colors. Particularly, we have 445 nanometer laser on this instrument. This is particularly good to do the chromosome karyotyping. We also recently acquired Bigfoot Spectral cell sorter. We're still under the validation process. This sorter equipped with 7 lasers up to 60 parameters. Generally, more lasers a particular instrument has, more detector it has, it can look up more colors at one time. So how to book time on our cytometers? And to book sorting, please email to cellsort@mrc.lmb.com.ac.uk, and to book analysis time, please use Cario Connect. This is what Cario Connect looks like. So apart from the cytometers, flow cytometers we have in LMB, we also have other types of cytometers around us. We can get access to those cytometers subject to a charge. If you feel you are interested in using these cytometers, please contact us. So there are another type of cytometers called image stream. So this is basically a combination of flow cytometer and a microscope. So it takes the pictures as the cell passes through the flow cell. It requires the fluorescent intensity of each cell, or a combination of the fluorescent intensity in the cell if you use more than one color. So there are mass cytometer and image mass cytometer in the CRUK Flow Cytometry Facility. So I'm not going to go to the details of this. It's totally another chapter of cytometry. Basically, rather than use fluorescent dyes, it use the antibody tagged with metal isotopes. So these discrete signal that coming from measuring the mass of each metal can allow the capability to look up to like 40 plus markers at any one time. Also, the image mass cytometer on top of standard mass cytometer, also they coupled an imager onto a standard mass cytometer. So basically, they can offer the spatial resolution when you want to look at tissue sections. So today I'm going to concentrate the basic concept of flow cytometry and what they can do. So the first, I'm just going to talk about the definition of flow cytometry. So the flow cytometry is a laser-based technology that used to analyze the characteristics of cells or particles in the fluid system as they pass one by one through a sensing point. It measures fluorescent intensity produced by fluorescent labeled probes. So that detect proteins or ligands that binds to a specific cell-associated molecules, for example, DNA. Flow cytometry analyzes the expression of cell surface and intracellular molecules. It can characterize and define different cell types. Analyze cell size also allows simultaneously multi-parameter analysis of single cells. So compared to a microscope, what is the advantage and disadvantage of a flow cytometry? So we can see clearly the microscope can tell you the location of antigen, which cytometry cannot. However, the biggest advantage of flow cytometry is the throughput. So it can analyze many cells in a short time frame. Also can look up, also can measure at numerous parameters at once. So as we can see here, on the right, there's a dot plot, each dot representing a cell. So depends where the cell locates. It can be single positive for a particular marker or double positive for two of the markers. So how does a flow cytometer work? So I'm going to go through the fluorophores and the fluorescence, and also give you a little bit more details regarding the components of a flow cytometer. So what are fluorophores and fluorescence? Fluoroforms are the molecules absorb a light energy at a given wavelength, emits at a longer wavelength. For example, the fluoroform fluorescein absorb an external light, and the electron of the fluorescein gets excited and rise to the maximum energy state, and then they move to a lower and more stable energy state. Eventually, it returns to the ground states, releasing the remaining of the energy and releasing a light. So this, when they're releasing the remaining energy, they will give out a fluorescence. We call it emission. Although the absorption and emission of a given fluoroform covers a wide range of the wavelengths. However, the difference between the max of emission wavelengths and max of excitation wavelengths is called Stokes shift. Stokes shift is used in flow cytometry. So there are many forms of, there are several forms of dyes. So the simplest one is a single fluorescent molecules. As showed in the table, so there are a range of single dyes we can choose from. They are excited by different wavelengths. They are emits at different wavelengths. You can use them singly, or you can use them in the combination. Depends what's the emission light is. So they also classified the brightness also classified from 1 to 5, as the 5 as the brightest. We can see the PE is the brightest dye in this single fluorescent molecule form. So we have another type of dyes called tandem dyes. So tandem dyes derived from single dyes, which take advantage of Fred. So the acceptor molecule covalently coupled to the donor molecule. So the emission wavelength of the donor overlaps with excitation wavelengths of acceptor molecule. So the donor molecule transfers the energy to acceptor molecule that gives out emission light. So there's another table shows you the tandem dyes were usually used in flow cytometry panels. So for the tandem dyes, they often quite sensitive to light, also the fixation procedure. So it depends what kind of experiment you want to do. So you need to choose the dyes wisely. So another question is why we use tandem dyes? What advantage of this? So the tandem dyes are the clever ways to increase the Stokes shift, allows more detection, allows the detection of many more fluoroforms excited by a single laser lines. So these are the example of the fluoroforms we can purchase on the market. So the upper rows are all the range of the dyes. They all can be excited by a single laser line, 4 or 5 nanometer. So in this range, the BV421 and BV510 are the base molecule. That's the single fluorescent molecule. So the dyes BV605 up to BV785 are the tandem dyes based on the BV421 and BV510. So similarly, the BD has a range of brilliant ultraviolet dyes. Again, the BUV395 is the single fluorescent molecules. So the other dyes are the tandem dyes based on the BUV395. So by increasing the Stokes shift, we have many more dyes to choose from. So it can increase our panel that we used to stain our samples. So another important fluorescent molecules is fluorescent proteins. originally the fluorescent proteins are found in the nature. For example, the GFP are found in the jellyfish. And then later, these fluorescent proteins are modified and validated, used for intracellular markers in live cells. So this table shows the common fluorescent protein we are used in our flow cytometry experiments. These fluorescent protein can be used on its own or in a combination. So they're here in LMB, some scientists created the confetti mice that has up to 4 fluorescent proteins in their mice. So they are coding for example, coding for transcription factors that could report the expression of their nuclear protein. So I move on to the components of a flow cytometer. So the flow cytometer composed of fluidic system, lasers, optics, and detectors and electronics. So break down to each components. So the fluidic system of a flow cytometer is to transport cells or particles in a fluidic stream to the interrogation point of a laser. Ideally, the cells should be transported in the center of the laser beam, past the laser beam one by one. To achieve this stable running of fluid stream, flow cytometry utilize the principle of hydrodynamic focusing. Once the sample is delivered into a flow cell, the sample stream will be forced by the sheath fluid into the center of the stream, forming a single file. So the single cells or particles can pass through the laser beam at one by one at a given moment. Because the sample stream does not mix with the sheath stream based on the principle of laminar flow, when the sheath pressure is constant, we often want to increase the flow rate. So increase the sample pressure is the only way to increase the flow rate. However, increase the flow rate coming with consequences. So it comes in with broadened distribution, decreased resolution, and increased coincidence. However, in the flow cytometry, when you're running a flow cytometer, there are always the balance between how fast you want to run your sample and how well your population is resolved. So optics of a flow cytometer includes excitation source and the mirrors and the filters that collect emission lights. So on the modern flow cytometer, we use the solid state laser at a given wavelength. So in here, the instrument we have a range of the lasers from ultraviolet to the infrared. Each instrument has different combination of lasers. Again, please refer to our internal website for the specification of our instrument. So there are lenses placed in front of the laser that used to focus and shape these lasers onto your sample stream. Also, the optical mirrors and the filters are used to separate and direct the emission lights. The most important ones, the most important mirrors and filters are the upper two. So dichroic mirrors, for example, 540 nanometer dichroic mirror, that acting as a beam splitter. So any light longer than 540 that will pass through, and any light shorter than 540 will pass through, and any light longer than 540 will get deflected into 45 angle direction. So then the remaining light will enter another light path. So the bandpass filter usually placed in front of a detector So it only transmits a specific limited bandpass while rejecting all other wavelengths. The short pass filter and the long pass filter acting the similar function as the bandpass filters. So put these optical filters into a context to better understand how they work. So take examples if we have the cells that are labeled with Fixie and the PE dyes. Fixie emits maximum light at about 580 nanometer. The maximum emission of PE is around 575 nanometer. So we can see as the cell passes through the laser beam, the lights emitted from Fixie and PE gets collected by the collection length, and they enter the light path, encounter the first dichroic mirror 510 long pass. They both are longer than 510 long pass. They will pass through this dichroic mirror and encounter the second dichroic mirror. So on this mirror, Fixie signal is less. The maximum Fixie signal is shorter than 550, so they get deflected onto the it gets deflected onto the 530 bandpass, and the remaining light will pass through. So again, the PE will be PE maximum signal is shorter than 620 long pass. It gets deflected into the right angle and enter 585/40 bandpass. So by this way, we can separate Fixie signal from the PE signal. Now the flow cytometer field is moving very fast. So next generation of flow cytometers are spectral analyzers. So what are spectral analyzers? What's the difference from the conventional flow cytometer? So differ from the conventional flow cytometer, the spectral analyzers collects the light in a different way. So rather than collect a limited portion of the light, the spectral cytometer disperse a fluorescent signal of a fluorophore into a detector array. So all this signal from this detector array can give you a unique fluorescent reference that can be, so this particular signal can be unmixed from the fully extinct sample. So spectral analyzers offers more flexibility in terms of dye you use and the combination of dye you use. So take an example. So as we can see on this image, the conventional cytometer only collects at certain wavelengths of the light. And the spectral analyzers collects then the whole wavelength of the light. So take an example of APC and Ultrafluorescence 647. So on the conventional cytometer, we are not able to separate APC from Ultrafluorescence 647. However, on the spectral analyzers, because the APC has its unique fluorescent signature, and Ultrafluorescence 647 has its own unique fluorescent signature, so we are be able to unmix APC from Ultrafluorescence 647. Also, spectral analyzers can give you another advantage, that is autofluorescence subtraction. So on the spectral analyzers can detect autofluorescence and classify as a real fluorescence signature. So we can see here, take an example, there's a sample unstained light whole blood sample. In this sample, we have different population of cells that give out a different degree of autofluorescence. So before the autofluorescence subtraction, the autofluorescence, intrinsic autofluorescence, the autofluorescence is intrinsic, is not be able to move, is not be able to generally removed, identified by the conventional cytometer. So these autofluorescence could give out a false positive into and overlap with a certain markers you want to look at. By apply autofluorescent subtraction, and we can define the real positive of the cells, reveal the true positive for the markers you are looking for. So electronics of a flow cytometer acts as the brain. So the detector of the electronic system in a flow cytometer converts incoming photons to electric currents, hence voltage pulses. There are several types of detectors, and the different flow cytometer uses different detectors, but they actually acting at the same way. So these voltage pulses are further analyzed and digitalized and stored into the computer for the further analysis. So how the voltage pulse are analyzed? As we can see here, as the cell enter a laser beam, a voltage pulse rises from the baseline. And as the cell fully illuminates in the laser beam, the voltage pulse reaches peak. As the cell exits the laser beam, the signal starts decrease, eventually returns to the baseline, returns to the threshold. So the width of the signal is related to the time that the cells are passing through the laser beam. The height is related to the fluorescent signal. So the area is the integral of combined from the width and height signal. So how the data collected and analyzed. So we are, I'm going to talk to you about some parameters. The first parameter is the Fourier scatter. So the Fourier scatter is the light, is the light that's collected at the Fourier detection when the laser strikes cells. Although the Fourier scatter is directly related to the refractive index of the cell, they can give us some indications related to the cell size. For example, the small cell will reflect less light compared to the large cells. So the voltage generated from the small cells will be smaller than the large cells. If we plot the intensity of the light into the histogram, this will give us some information regarding to the distribution of the cell size in a given sample. So the set scatter collecting the light at 90-degree angle. So the set scatter related to complexity and granularity of the cells. So in a certain sample, and you can see the cells are grouped together according to the similar granularity and complexity of the cells. When we combine the two parameters together, we can already see clearly, we can clearly identify a different population of cells only from the scatter signal. So also don't forget, put your threshold into the right place because the cells come in a different size. So to review the signal we truly need, we need to remove the noise and the small cells that we don't need to look at. So take an example of a whole blood dot plot, whole blood sample. So from the Fourier scatter and the set scatter, we already can see, we already can classify the different population of the cells. So we can see the lymphocytes, monocytes, and the neutrophils. So how the fluorescent signal are collected and measured is similar to the fluoroscatter and Z-scatter of a fluorophore that attach to an antibody binding to the cells, and it emits the light when the cells pass through the laser beam. So the fluorophore emits the light. However, intensity of a light depends on a couple of factors. The first one is depends on the number of fluorophores that attach to your cells. For example, you have like weakly expressed protein, they will have less fluorophore attached to the cells, resulting in a smaller voltage pulse. Then you will have highly expressed protein on the cells, and that will result in the higher voltage from that cell. So we can plot the fluorescent intensity onto a histogram, so we can clearly classify our negative population and the positive population for that particular fluorophores. So in flow cytometry, where very often used is the 2D plot rather than only histogram, because we often use more than one fluorophore in our sample. So in this case, we can see by plotting a fluorescent parameter against the Z-scatter, we already can classify several populations of our cells. So the gates or regions are placed around of the cell population of interest, and these subsets can be further analyzed. For example, in this case, the red population is lymphocytes. If we gaze on the lymphocytes, we can further classify the lymphocytes into CD4 population and CD8 population. So all this data we acquired from a flow cytometer is saved as FCS files. So FCS files is created and maintained by the International Society for Advanced Cytometry. So there we have a third-party analysis software to use in our facility. There are FlowJo and FCS Express. If you need to analyze your flow cytometry data, please contact us. We can give you a license. So there are some statistical analysis. So there are some statistical tools that are used to analyze the flow cytometry data. So I listed some of the common statistical parameters you used to analyze in flow cytometry. For example, percentage of the total is the percentage of the population of the total events collected. Percentage of parents is the percentage of a population based on the parent gates. Median value usually used for the log scale, and a mean value is usually used for the linear scale. So the coefficient of variation is related to the standard deviation of your data. So we are not only analyze the cells, also we can sort the cells. So when we have a population of cells and we stain these different fluorophores, so these fluorophores, for example, we stain the population with 3 different colors, red, yellow, and green. So we can, if we want to isolate the particular population that labeled with green fluorophore, we can pass this mixed population of cells through a cell sorter. So we can put a gate around the population we want to sort, and the cell sorter will identify the particular population, apply an electrostatic to that particular cell population, and separate them, pull them out from the mixed population of the sample. So as we can see, we can sort the green population, and we can also sort the yellow population. And these sorted cells will subject to the further cell culture and analysis. So again, the Pierre Andre will give another talk on Thursday particularly focused on the cell sorting. This is an image from one of our cell sorters. So just to tell you how this cell sorter looks. So in the upper chamber, the nozzle will intersect with the laser beam and detect your cells of interest. And then there's a droplets will be vibrated. So the stream will be vibrated into the jobs. So we can sort up to 25,000 events per second, and the last detached job that containing your cells will be pulled away and sorted into a particular collection tube. We not only can sort into the tubes, we also can sort into plates. And also we can sort various containers according to the application, the subsequent application you're going to use. So let's move on to the sample preparation. So the sample preparation is key. In flow cytometry, a single cell suspension is necessary. So some of the cells come naturally in suspension, for example, spinocytes or cell lines. Some cell lines grow adherent, so that will need to be removed from plastic. We will need to use trypsin or acutase to remove the cells from the plastic, and the EDTA will help to make single cell suspension if your cells feel very clumped. So the tissues are more difficult. They need mechanical enzymatic dissociation. For example, use collagenase, and also we recommend to pass them through a cell strainer before you bring them to the sorting or analysis. The sample should contain as little debris and as few clumps as possible. So there are other considerations when you prepare your samples. So dilute your sample to a concentration the instrument can handle, and get rid of some unwanted cells by for example, by lysing your red blood cells if you use whole blood sample, and FICO your cells and separate mononuclear cells from other cell type. Also, it's important to include a viability dye to get out the dead cells. There are a range of viability dye, and there are some, so for example, DAPI, PI, and DRAQ7, and some other viability dye, they are fixable viability dye. They can be used together with your other fluorescence, with other fluorescent probes that you need, that requires fixation of your cells. So this is an example of using a viability dye. So the upper panel shows the population of cells I very routinely sorted in the lab. So the LC2 cells are classified as KRLG1 positive and ICOS positive. Before this final gate, we used a viability dye to remove the dead cells. So the plots, the lower panel of plots and showing the left plot showing without viability dye, and this little population that generated from that's basically from the non-specific banding from the dead cells. This looks like false positive. When you stain your cells, when you input the viability cells into a panel, we can get out the dead cells at first gate and remove this false positive and reveal the true positive of the population you are interested. So always including controls. So using the negative control to set your background value, use single color control to set your color compensation and the position of your positive cells. So there are basic principles in your panel design. For example, practice the dye on least expressed antigen, choose dyes with least spectral overlap, or use different laser for excitation. or choose the fluorophore that use different laser for excitation. So there's, because our fluorescence molecules, they have the wide range of the emission wavelengths. So that causes spectral overlap. Take examples of Alex Fluor 488 and the PE. So the 530 filters usually we use to detect Alex or Fluor 488 signal. However, the spectral from the PE can spill into the detector of Alex or Fluor 488. Verse versa, the 585 filter is for detecting the PE signal. However, the Alex or Fluor 488 signal spill into the detector of the PE. So you can see on this dot plot. So this is the Alex or Fluor 488 fluorescence. They not look Alex or Fluor 488 only. They looks like double positive of Alex or Fluor 488 and PE. So to solve the spectral overlap issues, we need to apply the compensation to our samples. How to do the compensation? To do the compensation, we will need single color controls. So for example, if we want to compensate Alex Fluor 488 out of a PE, and we want to compensate PE signal out of Alex Fluor 488, we will need single color. We will need each single color for Alex Fluor 488 and the PE. So we run the Alex Fluor 488 sample onto a cytometer, assess the signal spillover from Alex Fluor 488 to the PE, apply a compensation and correct the signal, pull this signal back to the Alex Fluor 488 channel. The verse versa, and we run the PE single color controls, assess the spillover to the Alex Fluor 488 detector, and then correct the spillover accordingly. So take an example. This is the peripheral blood was stained with CD4 fixate and CD19 PE. So we know CD4 is T cell marker and CD19 is the B cell marker. Without the compensation, we can see the upper panel, the graph C, the CD4. So this population looks a double positive, but we know the CD4 will not be double expressed with CD19. So after we apply a compensation, we can truly review our CD4 population and the CD19 population. That's the T cell population and the B cell population. So we move on to the applications and examples. We want to know what flow cytometry can do for your experiment. So the most simple example is fluorescent protein that used into the flow cytometry experiment. So the fluorescent protein normally can contact with a protein plasmid expressed in cell lines. In here, we have a BFP tagged protein. So we can see a clear negative population of the BFP positive cells that are reporting the protein you are interested. And also the other form of the protein will show the range of expression. The expression pattern is not homogeneous. So they have a range of expression level. However, by setting a negative control, you will reveal by setting the negative control, then you will see a true positive of GFP expression that reports to the protein. So the second example is the fluorescent protein are used in the genetically engineered mice. So in this mice, the 4 fluorescent protein are used that reports the transcription factor. So we can see the wild type has no fluorescent protein on it, and the Katushka only cells and tomato-only cells. And also we have a double expression of Katushka and the tomato. And BFP, so we also have BFP and TFP. TFP is the teal fluorescent protein. We can clearly see the single positive for BFP and a single positive for TFP, and also the double positive for both of the fluorescent protein. So flow cytometry can also use to analyze the apoptosis. So annexin 5, fluorescence tag annexin 5 is commonly used to assess apoptosis of the cells. So annexin 5 binds to a lipid protein. During the apoptosis, the lipid protein will be translocated from inner side of the membrane to the outer side of the membrane. So we stain the cells with protein with the PI and annexin 5. So the annexin 5 single positive will indicate apoptotic cells, and the protein iodide will stain the dead cells because they will enter the compromised membrane, stain the nucleus. So by this way, we'll see the apoptotic only cells and the dead cells. So also flow cytometry can analyze the proliferation of your cells using the CFSE. So the CFSE can enter the cell membrane rapidly and then gets cleaved by the enzyme in the cell. It will give out a fluorescent molecules. The fluorescent molecule will covalently link to the intracellular proteins that will track the cell division. So when the cell divides, the fluorescent signal from the CFSE will halve. And by tracking the decrease of the signal, we can clearly identify the division of the cells. So this is example to show the T helper cell proliferation from a mouse spleen. These cells are loaded with CFSE and analyzed on the day 4 under the stimulation of cytokines in culture. So we can see the T cells undergo a division, also a L13. The GFP reported L13 are analyzed together with the cell division. The divided T cells expressing L13 reported by GFP, indicating their development into Th2 cells. So we also can analyze cell cycles by flow cytometry using DNA dyes. So here we use the Hurst 3342 to stain the RPE cells. So the Hurst will integrate into the DNA, and on the cell cycle experiment is very crucial to remove the doublets. Because when the two G1 cells coming into the laser beam together, that will classify as a single G2 cells. So the first gate, we usually gate out the doublets from the, according to the peak and area signal, and plot Hurst in the histogram. We can see the first peak related to the G1 phase, and then the cells enter the S phase and enter the G2 phase. So this experiment also, Emmanuel also look at the cell surface marker related to the different stage of the cell cycle. So this is the example to show you that it's crucial to titrate your DNA staining. So this is archaea samples. Archaea cells are very small organisms that only have a circular DNA. So the upper panel shows you to use higher concentration of DAPI that in the microgram range, and the lower panel and show you the concentration of DAPI in the nanogram range. You can clearly see the lower concentration of DAPI will give you a better resolution, for the cell cycle analysis. So which does, so it tell you that not all the high concentration of fluorescent probes will give you a better resolution. So the titration is crucial here. So this is another beautiful example to show you the cell cycle analysis and in human myeloid leukemia cell lines. And there's a haploid cells and a diploid cells, and also diploid cells enter a cell cycle. So we can see the 4N indicates the G2 phase. So the we also can analyze the S phase in the cell cycle by using BRDU. The BRDU is a thymidine analog dyes incorporate into DNA as cells enter the S phase. You can see we can detect the BRDU by fluorescent labeled antibody. So we can clearly say here the G1 phase and S phase and the G2 phase. Some of the cancer cells normally undergo like some of the cancer cells have very abnormal amount of DNA. They will not undergo the normal cell cycle. So we can see here we have several cell cycles happening in one cell, and some of the cells are not be able to divide normally. So this is the paper published in Cell Reports by Zhang. They shows textbook style of cell cycle analysis. You can have a look if you are interested. So also flow cytometry can do chromosome karyotyping. So using this chromosome karyotyping using DAPI and the PI. So we can clearly see the flow cytometry can identify a different copy, can identify the chromosomes in the cell. So we can clearly see the Y chromosome, the X chromosomes. So immunophenol typing is a big thing in flow cytometry. Usually that will use the fluorophore conjugated antibody. This is a general workflow, how you stain your cells and apply to a flow cytometer. So it's important to titrate your antibody to find optimal concentration for use. Again, it doesn't mean the higher concentration of the fluorophore, the antibody, the fluorophore conjugated antibody you use, the better resolution you can get. So usually you will have to titrate your antibody before you start to design a bigger panel. So this is example from the fresh whole blood, and we can clearly see the lymphocytes, monocytes, granulocytes, and upper panel shows T cell markers, and upper panel use the T cell markers to classify a different subtype of T cells, and the lower panel shows you the granular cells and dendritic cells, B cells, and also their activation states by using HLA-DR markers. So this is an example to define hemopoietic stem cells. So by using the stem cell marker CKIT-SCAR1, we can clearly identify the common lymphoid progenitor and other progenitor cells. This data from the Huang. So there's another example to identify hemopoietic stem cells. So there are 3 ways to identify hemopoietic stem cells. And particularly want to point out this on the third column of the plots, and that shows the set population. The hemopoietic stem cells are splitting out the hash that's supposed to integrate into their DNA. So they result in decreased signal in hash. So we classify the site population and also define further with other lineage markers and stem cell markers such as SCA1 and c-KIT. So there's other applications we use. So there's other applications we can use cell sorting to do. So this one we identified and isolate the homostatic stem cells and isolate them and transfer them into the mice. So the mice, the bone marrow cells was harvested and also analyzed how the cells from the donor cells proliferate, what kind of cell type they are turned into. So we also can look at intracellular cytokines by flow cytometer. So this is the general workflow to look at intracellular cytokines. So here we're looking at intracellular cytokines L2 and interferon gamma with this T cell proliferation. So we can see the LC2 produced from the first cell division onwards, interferon gamma produced after a number of T cell divisions. So there's also, if you want to look at intracellular cytokines in the serum, also we can use the particle-based immunoassays. So we can multiplex, use the beads that are coated with different kinds of the cytokines. And also, so within each population of beads, we have a different intensity of the cytokine fluorescent markers. And we can apply these beads to the serum and pull out the cytokines that we want to analyze, and also apply for another layers of the antibodies to define what kind of cytokines they are in here. So this is another example to look at intracellular nuclear proteins. So Noe kindly laid out the transcription factors that he is looking at. So there's a range of transcription factors that they located in the nucleus, for example, GATA3 and the UMIs, TBETs, and the Ki67. So depends what kind of the antigens you want to look. Maybe you have to use the different protocols, especially permeabilization and fixation protocols, in order to clearly identify these proteins located in the different compartments of the cells. So flow cytometry also can analyze calcium influx by using a dye called INDO1. INDO1 is the dye, is a ratiometric calcium dye. When bounds to the calcium, it emits at 405, and when unbounds, it emits at 475. So by ratio out this, by the ratio, using UV 405 wavelengths over 485 wavelengths will give us a clear signal how the cells respond to the stimulation in terms of calcium influx. So this is the naive T cells. So this is the stimulation from naive T cells. You can see clearly the calcium enter the cell into the T cells and return the ground states. And the control is to stimulate the T cells by iomycin to clearly show the cells is functional. So we also can detect FRET by flow cytometer using the FRET pairs. In here, they're looking at the protein aggregations. So upon the stimulation, the FRET happens. So the CFP here acts as a donor fluorescent molecules, and YFP is the acceptor molecules. when the protein aggregates, the FRET happens, you can clearly see the fluorescence profile shift towards YFP from the CFP, indicates here. So this is another FRET experiment to detect membrane fusion. So when two cells are fused, the DIO labeled cells, so the DIO labeled cells fuse with DI labeled cells. When they fuse, the FRET happens, and we can clearly see the population from here. That's the signal profile shift from the DIO to DII. So I'm not going to go into the too much details how you perform the FRET experiment. If you are interested, please again contact us. So I'm simply show we are not only can analyze cells, we also can analyze bacteria. Because the bacteria are much smaller than the cells, we have to pay attention to how to pull the bacteria signal out of the noise. As here, you can see here that a bacteria signal are overlapping with the noise. So again, contact us to if you want to analyze bacteria, we can help you to set up the instrument. So the last one is affinity assays, and this is the example to show in the nanobodies that binds to the antigens. So first round, we can select the weakly bound. We can select a nanobody that weakly binds to the antigen, and then these population will be amplified and incubate with the antigen again. You can see from round 1 and round 2, we already enrich the population of the nanobody that can bind to the antigen better. So there are some references books if they are interested to read, and also some reference website reference to if you want to look at further in terms how to use flow cytometer. So please refer to our internal website for more information. And again, come and talk to us regarding the training, self-sorting, flow experimental design, or you want some advice on the fluoroform. Anything else, you're welcome to talk to us. Thank you. Oh, finally, there are some of the flow cytometry clubs that around regionally and in the UK and worldwide. Thank you very much. If you have some questions, you can put into the chat. Or so what types of factors would determine the recommendation of one analyzers over another? Yes, so there are several factors. One factor is what type of the sample you want to analyze. So generally, the people use cells. So all our cytometers are good for analyze cells. Another important factor is about how many colors you want to look up at one time. So some of the instruments has more lasers, have more detectors. They obviously will be better to look at the more colors than some of the instrument has less lasers. So it depends how many colors you want to look. Also, what kind of dye you want to use. Some people use brilliant UV dyes that definitely requires UV laser. So some of our instruments has UV laser, some instruments doesn't. So yeah, so come to talk to us and tell us what you're going to do and how many colors you want to do, what exactly you want to do. And we can choose the right instrument and train you on that instrument as well.